everyone. Welcome to the Arthritis National Research Foundation's Researcher Spotlight Series. My name is Aisha. I'm the Development Coordinator, and on behalf of everyone at ANRF, I'd like to welcome you to our Spotlight on Rheumatoid Arthritis. We are so excited to have so many of you joining us. Before we get started, I have a few pieces of logistical information for you. This webinar is being recorded, and we will have it available on our website and YouTube channel in the coming weeks. If you have any questions, please type them into the Q&A. If we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. So to get us started, we'd love to hear from you. I'm going to launch a poll, and I want to know what brings you here today. Are you living with rheumatoid arthritis? Are you a caregiver or a researcher? Be sure to put that in so we can see who's here with us. So it looks like most of y'all are patients, and that's a great place for us to begin. What we'll learn today will help us understand what's coming next in terms of diagnosis and treatment. I'm excited to turn the mic over to our patient speaker, Stacy Stringer-Bayshart. Before Stacy ever thought about hospitals or infusions, she was a varsity athlete being recruited to play college volleyball. Then, in the span of a few months, her body changed dramatically. Pain, swelling, and loss of function derailed her plans, and for a long time, no one could explain why. So today, she's here to share not just her diagnosis, but her lived story of adapting, rebuilding identity, and learning how to live a meaningful life with a chronic illness. Stacy, take it away.
Thank you so much, Aisha. So my story started back in 2004. I was a senior in high school playing competitive volleyball year-round. And in my time off, I worked at a local coffee shop. This is where I started to experience horrible pain and swelling in my hands. It got so bad that I would drop hot steaming milk all over myself. So this sparked my family to say we gotta head to the doctor. And I stumped the doctors at the time. I was 17 years old and experiencing something of an 80-year-old. So because I was a teen, they thought I was spending too much time in front of a screen in my phone, and they were sure I had carpal tunnel. And if you've ever been tested for carpal tunnel, it is extremely painful. But this went on for months. No one could figure out what was going on. My pain got worse, and they just upped my dose of prednisone. Volleyball became extremely painful to play, and this was devastating for me. I had dreams of playing college volleyball, and I would collapse on the floor after I made a hit because the pain in my knees was so bad, and I couldn't open my hands to hit a ball. So my family and I had a very long talk about having to turn down my 2 college scholarships and stay in state in Oregon. And this way I was able to go with my parents to doctor's appointments and continue searching for what diagnosis that I had. I remember, though, at college, calling my mom in the morning crying from my dorm room because I spent the night freezing cold because my comforter felt like it weighed 50 pounds trying to pull it up over me. And I was so frustrated. It had been over a year with no diagnosis. And finally, someone mentioned, why don't you have them check your inflammatory markers? And sure enough, that was it. I was diagnosed with rheumatoid arthritis, and it felt like a weight was lifted off of my shoulders just knowing the name of what it was. But going into my first rheumatology appointment, there were some things I wasn't expecting as a 20-year-old with my whole life ahead of me. I was told I couldn't have kids and I couldn't drink, which to a 21-year-old was devastating. But there were caveats to all of that with medication changes. Things were possible. But the thought of maybe not having kids was so sad. So as a young adult learning about this diagnosis, it was heartbreaking. But I persevered through all of it and found a way to get through college with this new life experience. So after college, I wound up moving up to Seattle and started a career in healthcare communications. My first job, funny enough, was actually working with RA patients to tell their stories in marketing and advertising. But it was so nice being able to relate to others. But it was also hard trying to balance work and managing the pain I was in. It's definitely different working 8 hours a day with your brain functioning and also trying to manage that up and down of achiness. But during the 10 years that I lived in Seattle, I found a really great rheumatologist up there. And I felt like I was chasing feeling better. I had run through a number of different biologics. I felt like if I didn't switch to a new one every year, it was every other year. And finally, my rheumatologist helped explain to me that I may never actually feel as well as I did before I was diagnosed, but if I felt at least 20% better, I needed to consider that a win. And that really put things into perspective for me. I wish she would have told me that sooner because I wound up blowing through about all of my options in the 10 years that I was in Seattle. It was crazy to think about it that at the age of 32, I had no more treatment options available. My RA got really bad in 2018, and I could no longer live on my own. I had to move back to Oregon and move in with my parents. Thankfully, I was able to get a job at my family business, and that gave me the bandwidth that I needed to focus on my disease while also taking care of what I needed to. I did have a close family friend tell me and really push for me to find a doctor that was outside of my local physicians. And that's when I applied to be a patient at Johns Hopkins. I found an amazing doctor there, and it's something that I always push for other patients when I speak to them, to really fight for a doctor that they get along with and that understands your goals as a patient. Mine was I needed to step outside of the box and find something that was maybe research driven and really find a new treatment. So I found a great doctor, and he pushes the envelopes with the insurance companies and helps really fight for me. And I'm proud to say that after 5 years, I've been on the same medication and have a treatment that's working for me. But this is why ANRF and the work that the researchers are doing means so much to me. Though we may not see the work they are doing come to fruition immediately, I know that the work they are doing will directly affect my quality of life and many others' quality of life in the future. And for that, I'm incredibly thankful.
Stacey, thank you so much for sharing your truly incredible and inspiring story. Now, I'd like to introduce Dr. Renuka Nayak, a physician scientist at UCSF. She cares for patients with autoimmune diseases like RA, lupus, and psoriatic arthritis. Her research is focused on the gut microbiome, which is the bacteria and other microbes living in the digestive system, to see how it affects disease and treatment. Today, Dr. Nayak will share some background information on RA and how it can affect us. Dr. Nayak?
Thank you, Aisha. I'm so grateful to be here with you all as a physician and a rheumatologist, and also a researcher that's been supported by ANRF. And so I'm just going to go through 4 major categories about rheumatoid arthritis, like a very brief overview, some of the comorbidities, the symptoms that patients present with, and then treatment options. But I'm going to keep my remarks brief so that we can hear about the exciting cutting edge research from our researchers. And I will be there at the panel at the end to answer any additional questions. So rheumatoid arthritis is an autoimmune disease, which means that the immune system is kind of not behaving normally. But we don't know what the cause is, and it's likely multifactorial. So a lot of things are probably pushing a patient towards rheumatoid arthritis. That includes environment, genetics, a multiplicity of factors. It affects about 1% of the American population as well as the global population. And because the immune system really travels throughout the whole body, right? It's there to protect us from viruses and bacteria, among other foreign agents. It travels throughout all parts of the body. when something's wrong with the immune system, it can affect all parts of the body. So even though rheumatoid arthritis has the word arthritis in it, it's actually a systemic autoimmune disease that can affect multiple organs. And so a lot of my patients are surprised to learn that. Because, I ask them, do you have eye symptoms or lung symptoms or skin symptoms like rashes? And it's important as a patient to understand that it's not just an arthritis, it's a systemic autoimmune disease. A lot of time, patients initially present with pain, swelling, heat, and redness of the small joints of the hands and the feet. And so that could be like the initial manifestation for a majority of patients. I have in my career seen patients actually present with lung symptoms where the rheumatoid arthritis actually didn't present with arthritis, but it presented with inflammation of the lungs or inflammation of the eyes. That's very rare, but it does happen. And it underscores the idea that like it's a systemic autoimmune disease. And that, I always tell my patients, we want to look for everything, right? And that's why you need a rheumatologist who's looking for these things, for these symptoms in different organs in patients. If rheumatoid arthritis is not treated quickly, the immune system will destroy the joints or other affected body parts. Including the lungs, the joints, sometimes the heart, the eyes. And so getting in with a rheumatologist, making a quick diagnosis and getting treated quickly are really important. Timing is of essence. And I'm going to, again, talk about treatment options a little bit later. We don't have a cure, but that's why ANRF is here to support these early stage researchers. But we do have a lot of medicines. When you heard a little bit about that from Stacy, that, even within a lifetime, actually, the number of treatments have really expanded in the past couple of decades. It affects women more than men. About 2 times as many women as men are affected, but men are still affected and sometimes have worse disease manifestations like lung disease. And as you heard, like the disease can affect young patients like Stacy. It can present early in the 20s and 30s, but it can also present later in life, like the 60s, 70s, 80s. And so there's kind of like these two peaks during a person's life where they are at risk for rheumatoid arthritis. Common comorbidity, so this is the second section. The rheumatoid arthritis can sometimes co-occur with other autoimmune diseases like Sjogren's or lupus. When a patient has lupus and rheumatoid arthritis, we call that rupus. Our patients tend to have cardiovascular disease at an earlier stage in their life. And so you want to be talking with your rheumatologist and also your primary care doctor about like, are you doing things that are good for your heart health? Because cardiovascular disease ends up being something that affects the lifespan of a rheumatoid arthritis patient. RA patients are also at higher risk of osteoporosis. All that inflammation in the body is causing the bones to lose their mineralization, and patients are at increased risk of hip fracture or spine fracture. So you want to be talking with your rheumatologist, not just about your joints, but also the health of your bones. And then, interestingly, rheumatoid arthritis, it's a problem with the immune system. So actually, there's also an increased risk of infection that's independent of the immunosuppressive medications that we're giving. And so that's another reason we want to kind of control the immune system and get people back in line so that their immune system is also better prepared to resolve infection. RA patients also have an increased risk of cancer, and then other inflammatory diseases like periodontitis can co-occur with rheumatoid arthritis. Okay, so the symptoms. This is the third section. Patients will often present, like one of the first manifestations is they're feeling bad. They're feeling achy. We heard a little bit of this from Stacy's talk. They have fevers, they can have weight loss and fatigue, and they just don't really understand what's going on. They may wake up in the morning and feel stiff. Now, I wake up in the morning and I feel stiff, but after about 5 minutes, I'm like up and ready to go. But my patients, when they have active disease, they'll say it takes me about an hour to loosen up the joints and get as good as I'm going to get. And so that's abnormal to have an hour or more of morning stiffness. As I mentioned, you'll have joint pain in the small joints of the hands and the feet, but it can affect other joints as well. A lot of our patients, after they've had disease for a while, will also have their knees affected and need knee replacements. As I mentioned, like if this immune inflammation goes unchecked, it can lead to joint deformities and impaired function. You heard Stacy talk a little bit about like how it affected her sort of plans for volleyball and her future career aspirations. I've had, bakers who are affected, they can't make their cakes, or musicians can't play their instruments. And so it's really important to control your arthritis so that you can improve the livelihood of patients. And then, as I mentioned, like patients can also present with non-joint symptoms like difficulty breathing, cough, not being able to exercise for as long as you normally do, eye pain, redness, rash, dry eyes, dry mouth. Okay, this is my last section on treatments, and I'm going to make it brief, but early diagnosis and treatment is so important because we have this window of opportunity. And back in the day, it used to be only gold and aspirin that were available for rheumatoid arthritis patients. But now we have a number of different treatments that I'm not going to go into because it would take a lot of time. Patients who have very mild disease get started on a hydroxychloroquine, but most patients actually, by the time they present to us, they have moderate to severe disease, and almost all of them get started on oral methotrexate. That's the focus of my research. And about 1/3 of patients will do well just on that one therapy alone. So it could be a good drug for about 1/3 of patients, but the remaining 2/3 will need additional therapy added on to the methotrexate. And those include conventional synthetic DMARDs, disease-modifying anti-rheumatic drugs, and also biologics that target particular cytokine pathways, parts of the immune system. And this has just been tremendous to observe, even in my lifetime, the drugs that have been developed because of research that's supported by foundations like ANRF and also the NIH. And so even despite all these therapies, we still have patients that are not adequately controlled and have kind of gone through all of the treatment options, like as we heard from Stacy. And that's why we need additional research to find novel therapies. I will also, just because I study the microbiome, will also remind everybody there are also non-pharmacologic therapies available to rheumatoid arthritis patients. So, Stacy mentioned, she wished she had kind of changed her thinking about what to expect from the disease. So, making sure you see your rheumatologist every 3 months, thinking about things like cognitive behavioral therapy, physical therapists to help you figure out how to pace your activities in the day, find adaptive devices to work with to make your lives easier at work and at home, thinking about sleep, diet, and then also self-management. Taking agency of your disease process has been helpful to the patients that I've seen in my clinic. And then I think I mentioned diet, and then also making sure that you're protecting against infections, making sure you're up to date on your immunizations, your heart health, bone health, and then making sure you're getting your regular cancer screening. And with that, I'm going to end there and turn it back to the moderator.
Thank you so much, Dr. Nayak. That was a great and super informative intro to RA. Before I introduce our next speaker, I just want to remind everyone to please put your questions in the Q&A box. We'll have a few minutes at the end of the webinar for you to get any questions you have answered by our experts here. On that note, I'm excited to introduce Dr. Winter. Dr. Deborah Winter leads the Winter Lab of Macrophage Genomics at Northwestern University, where her team studies how immune cells called macrophages contribute to aging, inflammation, and autoimmune disease. By combining lab experiments with genetic analysis, they are trying to uncover why diseases develop and progress differently in every person. Her ultimate goal is to use this knowledge to guide more personalized and effective treatments for patients. Dr. Winter.
Thank you very much. I'm glad to be here today. And I want to start by first off thanking Stacy for sharing her amazing story. I also want to thank the ANRF for inviting me to share my research with you today. Okay, let's see. Bear with me. I did put in a few animations that might be a bit slow, but I'll do my best. Okay, I'd like to start by telling you a bit more about RA, although a bit more after Dr. Nayak's coverage is probably redundant, but bear with me and we'll get to the good stuff. So RA is a complex variable disease. Okay. It's classified as an autoimmune disease, which means that it's the result of the body attacking itself, particularly the immune system. This is unlike other forms of arthritis, like osteoarthritis, which are more of a wear or tear, can happen after injury. Oops. The primary presentation of RA are swollen joints that can be painful, stiff, but this presentation can be variable. Some patients may have many joints involved, some few, some may progress quickly, some slower. There's a lot of variation in how patients present and what other complications are involved. As mentioned, patients with RA have an increased likelihood of heart disease, Some may present with fibromyalgia, including centralized chronic pain and beyond what happens in their joints, and also a general fatigue. And also, some patients may experience other complications like in the lungs, skin, eyes, or nerves. RA is widespread across the world. It's estimated to affect up to 1% of the worldwide population. It typically presents in people between ages 30 and 60, though you can have some much younger, early onset RA, or much older, called late onset RA. Women are actually 2 to 3 times more likely to develop RA than men. And also, we don't know the cause. We think it's a combination of genetics, which is what you're born with, and environmental exposures, such as smoking or diet. So this means that certain individuals or families may be more likely to get RA, but it's hard to predict who actually will. So for example, sorry, was that a pair of identical twins who have the exact same genetics, one may get RA and one may not based on their environmental exposures. So this combinatorial causation makes RA challenging to predict and to treat. Another challenge with treating RA is deciding what treatment strategy should be given to a particular patient. RA is typically diagnosed with physical examination, which includes assessment of tender and swollen joint counts, blood work, which can pick up certain inflammatory markers, as Stacey mentioned, and sometimes imaging as well, such as MRI or ultrasound that can detect even subclinical inflammation. Lifestyle changes can help, mostly the general things you do to keep healthy, like have a healthy diet, exercise, weight loss, reduce stress. But RA is commonly treated with something called DMARDs, disease-modifying antirheumatic drugs. And recently, there's been amazing progress in developing biologics as a class of DMARDs, which work with the immune system to treat inflammation. However, a given DMARD may work in less than half of patients, and it becomes up to the doctor to use their experience and their instinct to decide on the best treatment course. The result is that the process is very time-consuming and painful and costly to the patient, and it creates a sort of trial and error system where one DMARD is given after the other until one works. It's really important, a critical need, to find a data-driven, personalized approach. But to do this, we need to have a better understanding of the disease. So, spoiler alert, that is what we are trying to do. All right, so a few things to know about the underlying biology of joint inflammation and the different cell types involved. RA affects synovial joints like the wrist, elbow, knees, fingers, etc. These joints have synovial tissue around where the bones and cartilage meet and synovial fluid inside. The synovial fluid is made up of 2 layers, a synovial lining, which is made up of fibroblasts and macrophage cells that kind of form this wall over here. I don't know if you can see my cursor or not. And the sublining or interstitial layer contains a whole mess of different cells. They form a cushion, they contain the blood cells, and there's also a small amount of immune cells, including macrophages, that are keeping things clean and clearing up debris. However, in arthritis, you get circulating immune cells from the blood entering the tissue and causing inflammation, leading to a chain of events, which is called synovitis. And this eventually leads to a thickened synovial lining, degraded cartilage, and bone erosion, and a very painful situation. Now I'm going to switch gears a little bit and tell you how I ended up studying RA. So first off, you should know that I grew up in Canada. I went to the University of Toronto where I studied something called computational biology, which is basically where we use computers to do biology. After that, I attended Duke University and did a PhD where my thesis focused on gene regulation. Following that, I went to Israel and did a postdoc at the Weizmann Institute, where I was first introduced to immunology, and I really liked it and how you can use genomics to study immunology. It was a great match. It stuck, and then I was recruited to Chicago, to Northwestern, where I established the Winter Lab of Macrophage Genomics. I took a particular shine to macrophages during my postdoc, and I wanted to learn computational approaches or apply computational approaches to use more about them. So on the right here, you can see a picture of me in the lab, but I'm almost never there. I mostly can be found in my computer, hence computational biology. And here's a picture, a somewhat recent picture of my current lab members. I think they're a pretty good looking group. So the goal of my research program is to use functional genomics to study how immune cells are programmed and so basically how the cells know what to do, when to be on surveillance, when to attack, what to protect, and what to do in different circumstances. Things like that. I'll talk more about what functional genomics is in a bit. So my lab combines a wet lab where we do experiments using both mouse models and patient samples. Then we generate a whole bunch of data. And then this is used in the dry lab. We use computers to analyze this data and model it and find patterns. We study macrophages because they can be found in nearly every tissue of the body. They help maintain health, but they also drive inflammation. And so they're basically involved in every disease you can think of. So the so I wanted to say that macrophages are very sensitive to their environment and responsive, so they can reflect the disease state and the health of a system. So by profiling macrophages, we can learn a lot about a patient's disease, and our goal is to use this information to inform clinical decision making, which basically means give doctors the information they need to make the best choices about how to treat their patients. Okay. So, as promised, I'm not going to explain the whole functional genomics deal. So, as you probably know, every cell of your body has a complete copy of your DNA. Here, I'm just showing one half of a pair of chromosomes, but humans really have 23 pairs of chromosomes. This is just for the picture. So, your DNA sequence is unique to you, and it determines how your gene looks. Your genes look, come on, okay. But different cells will make different copies and different combinations of genes. So here in this one cell, it's got 3 copies of this black gene, which we could imagine as a housekeeping gene that does everyday things like making the cell membrane and making the cytoskeleton. And it's got 1 copy of this green gene. So in our sort of imaginary situation, we imagine that this will turn the cell into a macrophage. On the other hand, if instead of a gene copy, it got a copy of the blue gene, it might be a totally different cell. But there's more to the genetic story because another person will have their own unique DNA sequence that is largely, but not entirely the same. So I'm showing it by using another shade of green, and we'll call this first light green person Alex, A for Alex, and the second person Bruce, B for Bruce. All right, so Bruce has his individual DNA sequence, and I'm showing it in dark green. So that it means that his version of the macrophage gene is a little bit different, but not so different that it doesn't still produce a macrophage that does basically the same thing normal macrophage functions. But there's even more to the story. The genetics that you were born with does not complete the story. Over time in your life, you're exposed to different factors in the environment, like based on your lifestyle, pollution in the environment, germs that you're exposed to. So in this case, Alex was affected by his environment, but it didn't really change how the copies of his genes, so his macrophages are the same. Bruce got an extra copy of his dark green gene, but it wasn't enough to make any problems. On the other hand, it might happen that Bruce is exposed to something worse, like he was a smoker, he got a specific infection, and this might be enough to create a problematic version of the gene. This gene can lead to a macrophage that drives inflammation instead of promoting health. Then Bruce has a problem. But the good news is that with functional genomic technology, we can read these changes in the gene copies. We can isolate a relatively small number of cells from a person's body and see what gene copies he or she has. Functional genomic profiling reflects inflammation on the genetics, as I've shown, and also on your environment. So it goes further than showing you what you're born with and gives you this extra look at what a patient has been exposed to. And this is key for disease like RA, which is driven by both factors. Also, your DNA sequence is largely static across your whole life, so it can't be used to show like treatment response or disease progression, but functional genomic output can. So that makes it very important. We call it functional genomics because we not only profile in individual states, but we also try to understand how these differences in the genes lead to functional differences in how the cells function and how they malfunction in disease. We can compare cells from healthy people to people with RA, for example, and investigate disease mechanism and causes. And we can also compare RA patients or any patients to each other and get a sense of how they vary and who will respond to certain treatments, and what genes are associated with response to therapy. So this is the key. This is what we call, I'm so used to just clicking, precision medicine, which collects data on the one hand. Oh, let's back up just a bit. Okay. Which collects data on how patients have responded to treatment in the past. So that's a data-driven portion. And then determines the best course of a treatment based on their individual factors. So that's the personalized part. And in this case, the thing we're reading is the functional genomic profile. One of the challenges to functional genomic approaches is picking the right cell to study because the different cells of your body will look different. But we think we have a good idea on where to start. And you may have guessed that it's macrophages based on the fact that I've mentioned them like 30 times already. Let's go. Okay, so bear with me. This slide has some data on it, but I'll walk you through it. So my lab studies macrophages specifically, which, as I've mentioned, are basically found across your body from your lung to your brain to your heart. And they're important in maintaining healthy tissues, and they're also involved in immune response and inflammation. One of the things we do to study macrophages is use mouse models. For example, a mouse model of rheumatoid arthritis called STIA. And using these models, we can test different parameters. We can change the genetics of the mouse. We can change their treatment. And we can see how this influences macrophages and how it leads to changes in disease, arthritis. So in our initial studies, we isolated macrophages from the inflamed synovial tissue of an arthritic mouse and compared it to healthy macrophages from healthy synovial tissue. So here comes the data. So if you look at these figures, we used a technology called single cell, where individual cells, so individual macrophages in this case, are profiled. And in this plot, each dot represents a different macrophage cell. What we found is that macrophage cells can be grouped into 4 types, which we've colored differently here, and that the proportions of these macrophages change between health and arthritis. So in the healthy joint, you got a lot of these green macrophages. So in particular, there's the dark green ones that form the synovial lining and the light green ones which are found in the interstitial space or the sublining. And then there's a smaller number of these light purple ones. In the inflamed joint, the numbers of healthy green macrophages are greatly reduced. And instead, there are many more purple macrophages, especially these dark purple macrophages, which we have learned have recently entered the joint from the blood and can be particularly problematic. These results were a validation that macrophages are important to the development of arthritis, and that knowing the composition of different macrophages is likely to shed light on disease. Okay, so we found that macrophages actually vary across RA patients. So the way we did this is we performed synovial biopsies on the inflamed joints of a cohort of patients with RA, and we isolated the macrophages. In case you were worried that synovial biopsy may be painful or worsen disease, we actually polled the first 31 patients that were involved, and they reported no change in their pain, stiffness, or swelling, and most of them agreed that they'd be willing to come back for another biopsy, which is great because this is how we could look at treatment response. So anyway, when we looked at the profile of the macrophages from different RA patients, so we collected these RA patients knowing nothing about them, treating them as one big group. But as soon as we profiled their functional genomics, we realized that there were at least 2 groups of patients. So in this figure, you can see how similar each patient is to another. Each row is a patient, and the columns are the same patient. So every patient is compared to each other. And when it's orangey red, that means that those patients' macrophages are similar. And when it's green blue, they're not very similar. And you can see that we've drawn black boxes to indicate these 2 groups. The first group was larger and the patients in it were much similar, and it turns out that these were patients with relatively controlled disease. The second group was smaller and the macrophages were more variable, and it turned out that these patients had more severe disease. They had higher tender and swollen joint counts. So from this, we realized that you really cannot treat an RA cohort as a uniform group. Next, we wanted to look at the specific genes driving the disease. So in this figure, it's kind of similar to the one before, the one on the left with the rainbow coloring, except now the rows and columns are genes, and we're looking at how similar the genes are, if they're bright red, or how different they are if they're blue, and we're grouping the genes into these 6 what we'll call modules. And each module contains genes that act similarly across patients. So they're high in the same patients and low in the same patients. And then you can go back and in this middle figure, the orange and purple one, and you can classify each patient by their gene modules. So each row here is a patient, and if they have orange, that means they have more of this gene than average. If they have purple, they have less of this gene than average, and if it's white, it's about average. So you can see that each patient ends up with a somewhat personalized profile. And then we showed that these gene modules were actually related to how their disease looked. So people who had low levels of module 4 were those currently being treated with a biologic DMARD. And those that had high levels of module 3 were in an early stage of disease. So this overall, and this study was critical to establishing that there's a great range of variability across RA patients. They're not just orange. They're everything from gray to orange and everything in between. And also showing that functional genomics can be used to learn something about disease state. And this sets the groundwork for what we're doing currently in the lab. So here is what we are working on now. The data I already showed you is from an initial cohort called REASON, which was established by Harris Perlman, who's the chief of rheumatology at Northwestern. And it included 6 U.S. institutions that performed minimally invasive ultrasound-guided synovial biopsy, which is what I showed the data from before. So now we are working to use this data to train models based on baseline and follow-up samples from these patients that will predict treatment response to DMARDs, particularly biological DMARDs. So this means right now we're working on the best methods to identify patterns in this initial data and optimize machine learning algorithms or artificial intelligence algorithms. At the same time, we are collecting samples from a larger independent validation cohort. Using this validation cohort, we will test the machine learning algorithm by using the model to generate predictions and then comparing these with the actual patient outcomes to assess the performance of our models. If they're good, we may be able to bring them into the clinic, but more likely, we'll have to do some amount of iteration. We'll have to repeat the process. We'll learn something more. Often new information arrives. Sometimes we realize that our initial model was too noisy or too biased. Maybe our initial cohort needs to be expanded to better reflect the whole variety we see in disease, or maybe we let in too much noise and we what's called overfit this model. So it's often a very iterative process here as we perfect a model that we can actually take into the clinic and that doctors can use. The train and test assess loop, it's important that it's done as many times as needed because we're dealing with people's health and we don't want to put something out there that's not as accurate and precise as possible. Okay, so to summarize what I've shown today, and in case you fell asleep, the functional genomics enables us to understand how individual genetics and environmental exposures lead to disease. RA patients represent a diverse population that do not respond similarly to treatment. And while some macrophages contribute to disease development, others promote resolution. And part of what we're doing is trying to understand this balance and what it means, and also in the future possibly manipulate this balance to develop novel treatments. So functional genomics provides the tools for precision medicine. All right, so just to wrap up, I'd like to acknowledge the people in my lab who worked on this project and my collaborators at Northwestern Abroad, as well as the funding organization who supported my work. But most especially, I'd like to thank the patients who contributed their time and provided samples to support our research. Thank you very much.
Thank you so much, Dr. Winter. It's so interesting how you're able to use functional genomics to find better and more individualized treatments for disease. Before we move on, just a quick reminder to everyone tuning in. If you have questions, please drop them in the Q&A box and we'll get to as many as we can towards the end of the webinar. I'd now like to turn the mic over to Dr. Arandjelovic. Dr. Sanja Arandjelovic leads a research team studying how the body clears away dying cells in RA and how this process impacts inflammation and disease progression. Her work focuses on identifying which immune cells act as the cleaning crew in RA and what tools they use to remove these cells, or what happens when that process fails. By understanding these pathways, their goal is to uncover new treatments for therapies that could reduce inflammation and improve outcomes for people living with RA. Dr. Arandjelovic?
Thank you, Aisha. I'm also very, very happy to be here, and I thank all the attendees for joining this webinar. It's a pleasure to speak to you about our research. As Aisha mentioned, we're really interested in dead cells and how they are cleared, and I'm super happy that we're giving this talk in October because my lab is obviously really big on Halloween and all things creepy and dead. And so I'll start off by saying that actually cell death is really a part of life. So essentially, cell death is something that happens in our bodies every day. So as we're here speaking, cells are dying in our bodies. The type of death that is occurring in our bodies all the time is called apoptosis, and it's a non-inflammatory homeostatic type of death that participates in processes such as development. So, for example, as a frog here, a tadpole transitions into a frog, its tail is being removed by these cells undergoing the process of apoptotic cell death so that a frog forms. Or perhaps what's more easy to relate to is the formation of our fingers. So initially during the development, we're all developing our hands with interdigital webs, which are these purple parts in between our fingers. And in order for our digits or our fingers to fully form, these cells in between the fingers have to die through this process of apoptotic cell death and be removed. Similarly, apoptotic cell death is part of homeostasis. So in many tissues in our bodies, different cell types have different lifespans and they need to die and be replaced. So, for example, in the brain, our neurons are long-lived cells that persist pretty much throughout our lifespan. But another example shown here is the lining of our gut, which the entire lining of our gut is estimated to be replaced every 3 to 5 days. So that's a lot of cell death and turnover. And that's shown here. These red cells are the ones that are in the process of dying. Similarly, as we have immune responses or as our immune system develops, there is cell death associated with that. And this is true both in the thymus, shown here on the left, where our cells of the immune system called T cells develop. And as they're developing, a lot of them die in order for better cells, more tailored to specific pathogens, to develop. Similarly, on the right, the process is the same for other immune cells called B cells in the spleen in this case. So our immune responses actually involve a lot of cell death. And then finally, and perhaps least appreciated, apoptotic cell death is also part of tissue regeneration. And an extreme example is shown here in this organism called Hydra, which when transected actually undergoes this process called apoptosis-induced regeneration that yields 2 Hydra organisms. So obviously this is an extreme example, but regeneration is known to occur, for example, in our livers. So basically, I've just told you that we have cells dying all the time. However, if we take a look at any healthy tissues, we're actually very rarely able to see those dead cells. And this is because the dead cells dying by apoptosis are very rapidly removed in a process called efferocytosis, where effero stands from the Greek word effere, or to bury or take to the grave. And it's really a process of engulfment or phagocytosis, which is specific for apoptotic or dying dead cells that are removed homeostatically. Now, again, as I mentioned, this process occurs in virtually all of our organs at a different rate. And I'm really happy that Debbie already introduced them to you, but this process is actually performed by cells that are called professional phagocytes. And this is because they are really good at performing phagocytosis. And phagocytosis is really eating from, again, Greek phago. So macrophages, again, as their name would say, are really good at this. Macro, so they're big, and they're phages, so they're very good at eating. So they're basically big eaters. And these cells are present in virtually all of our tissues, as Debbie mentioned. So, for example, in the liver, they're called Kupffer cells, or in the brain, they're called microglia. But they are there as immune surveillors to really find those cells that are dying and clear them before they burst and cause problems. So, really, you can kind of view macrophages as undertakers. They are there as a cleanup crew that will remove dead and dying. And the process of efferocytosis is really taking the dead cells to the graveyard. So a macrophage, or in this case, our undertaker, is performing efferocytosis, which is a type of phagocytosis that's really ingestion of dead or apoptotic cells. And so you can kind of view a macrophage also as the graveyard. So it is both the undertaker and the final resting place for most of these cells. So, what's really important about efferocytosis in comparison to, let's say, how a macrophage eats a bacteria or a different pathogen or a differently injured cell is that this process is actively anti-inflammatory. So, as a macrophage eats a dying cell, a homeostatically dying cell by apoptosis, they actively secrete anti-inflammatory factors, which really tell the tissue this is normal, this is part of basically janitorial duties that happen all the time. There's nothing severe happening here. And so we're really interested in this process for this particular reason. Why should we even care about apoptosis or efferocytosis in the context of rheumatoid arthritis? So we've known for a very long time that actually apoptotic cells can be observed in the inflamed joints of human rheumatoid arthritis patients. And this was seen as the patients were either undergoing biopsies, as Debbie mentioned, or need to have joint replacement therapy, as Renuka earlier mentioned. And what's shown here is actually a picture of a synovial joint of a mouse that's subjected to arthritis. And all of these brown cells are apoptotic cells that are not seen in the healthy, normal mouse or human joints. And there's a little inset that shows that picture increased. So what my lab is really interested in understanding is, where do these cells come from? What is their nature? Is this the reason, the reason that we see them? Is it because there's increased cell death in the context of rheumatoid arthritis? Is it maybe because they are removed less efficiently? Maybe the macrophages are not as functional. And this is really where I think research from groups like mine is intersecting really well with research from group like Debbie's, so that we can compare what we see in our models with what Debbie sees using her functional genomics methods to understand really the macrophage biology and how they may be performing this process under tissue distress. So I mentioned already that efferocytosis is anti-inflammatory, so obviously we're really interested in trying to understand whether there are parts of this process that we can harness for new therapeutic development. And then finally, understanding how these dead cells are removed and what the responses to them are is really important for maximizing those therapies that are targeting this excessively proliferating tissue that forms in chronic rheumatoid arthritis. So a lot of the therapies are aiming to either stop proliferation of the cells or actively induce their apoptosis. And so understanding how macrophages deal with those cells is really important for maximizing therapy outcomes. So how do we do this? Similar to Debbie, we also use mouse models of rheumatoid arthritis or preclinical models of disease. We use one of the same models that Debbie mentioned, where we can induce arthritis in different groups of mice and then essentially measure their paws. And as you can see here, the size of the paws of mice subjected to arthritis really increases. And this is due to this inflammatory process that leads to this tissue swelling and inflammation. And this is what an example of that would look like in a mouse. So this is a paw of a healthy mouse compared to a paw of a mouse with arthritis. And I'm sure that you can appreciate that there's a lot of tissue swelling here. And this is how we basically measure the disease to know what are some of the things that we can genetically or pharmacologically change that could affect the outcomes in this particular model. So the kinds of questions that we ask are focusing on cells that perform efferocytosis. We also like to focus on macrophages because they are the best at performing efferocytosis, although other cells can contribute. So what are the tools that these cells are using? Is one tool the same as another tool? So is using a broom better or worse than using a vacuum, for example? So we want to understand that by using genetics and pharmacological approaches. Similarly, we're also really interested in what it is that the dead cells are releasing. So the process of cell death by apoptosis is itself anti-inflammatory. So as a cell commits to undergoing this type of programmed cell death, which, as I mentioned, is homeostatic and non-inflammatory, these cells release factors that also actively tell the tissue, hey, there's nothing wrong going on here, just stay calm. And so what we do is we also model some of these processes in vitro or in tissue culture. So we like using cultured macrophages, which, as you'll see here, are labeled in a red dye, which we can then incubate with dead cells, which are looking like these little ghosts. And as I play this video, hopefully, what you will see is that as a macrophage is taking up each of these ghosts, the displacement of this dye occurs so that you can kind of see holes in a macrophage. And these are all events where a macrophage has taken up these dead cells. And then I hope the second thing that you can appreciate from this video is that the macrophages are really good at this process. So here's one macrophage that has bound probably upwards of 20 different apoptotic cells. Similarly, macrophages are really good at removing what they have ingested. So in this case, you will see now that same macrophage, again labeled in red, that is now eating apoptotic cells that we can't see because they are labeled in a fluorescent dye that will only become fluorescent and it will be pink, hence a pink ghost, once that it's inside the macrophage and the digestion process has occurred. And so what you can see now is those same voids where cells are ingested starting to turn pink as the macrophage is starting the digestion process. And then if I can go back actually and try to play this video again, you can see that this one large void is turning pink and starting to turn smaller and smaller as a macrophage is digesting it. So macrophages are really good eaters, but they're also really, really good digesters. And so the kinds of things that we do in the lab is we target different proteins or genes that encode those proteins for deletion or increased expression to try to modulate how this process is occurring. We use different pharmacological ways to try and either boost or decrease this process to see how that will change macrophage responses. Conversely, we're also trying to understand what are the factors that are released by apoptotic cells, and can some of these factors also be used for potential therapies. And so with that, I'll just basically finish off and tell you that just like macrophages, my lab also really likes to eat. And one of the molecules that we study is called Elmo-1. So Elmo is our unofficial lab mascot. And then we like all cells, all things dead and dying, and even our dead skeletons are really hungry, just like the macrophages. So thank you. I'm really happy to address any of the questions that the attendees might have.
All right. Thank you, Dr. Arandjelovic, for sharing your research. If you have any questions for Dr. Arandjelovic or any of our speakers today, please drop them in the Q&A box if you haven't already, and we'll be heading right into our Q&A. So our first question is, how can we contribute to research efforts as patients or advocates? And this is for all of you, so feel free to just jump in.
I'm happy to start. So I think that if you have, as a patient about research that is happening at the, at the physician's office that you're getting your care from, ask about it. Ask like, what kind of research is happening here for rheumatoid arthritis? Is there a way for me to participate? Another strategy is to go on clinicaltrials.gov, where all of our clinical trials have to be registered, and you can see if there's something that's happening locally in your area. You can learn about research opportunities there. And then the third strategy is actually just to support research by, supporting organizations like ANRF, and also telling your congressmen and senators to support the NIH, because so much of these advances in rheumatoid arthritis research come from funding research endeavors.
I think that pretty much covers it up. Thank you. Thank you, Renuka. Yes, really talk to your doctors, and support organizations like ANRF, and other organizations that really work with scientists to work towards finding new cures.
Anyone else have anything to add? All right. Well, thank you all for being here. As we wrap up, I just want to say thank you to our fantastic patient speaker, Stacy. We so appreciate you being here and sharing your story. Thank you to our wonderful researchers for sharing their innovative work understanding rheumatoid arthritis. You can see firsthand the impact that ANRF has in supporting these incredible individuals in their quest for a cure. And of course, thank you to all of you for joining us for this great event. If you enjoy this webinar, please consider making a donation today by visiting curearthritis.org/donate. Every contribution helps. And remember to follow us on social media as well to stay connected. Lastly, we will be sending our live attendees a survey after the event. So your feedback helps us plan for future webinars. So please share your comments with us so we can continue providing important and relevant content to you. Thank you all for coming, and I hope you have a great rest of your day.